Great, thank you. Can you see my screen?
Sure can.
Awesome, excellent. So I am Heather Root. I'm in the botany department at Weber, and I am going to tell you about some work I've been doing with the Forest Service relating to using lichens to measure nitrogen and sulfur deposition in dry forests. So this is kind of a change of pace, but actually I saw some of the same kinds of things that Tabitha was talking about with spatial variability. So let's see. This is the basic outline of my talk, and we're going to start with why forests. The other talks we've been thinking about have been talking about human health, which is also super important, but forests. So forests are near our places where pollution is happening, where air pollution is happening. They provide important services for people. For example, there are places where people go for recreation, hunting, and fishing. They also are massive carbon stores. And when there's pollution in the air, like particulate matter or other things, it is over our forests and it lands on our forests when it comes out of the air. And that's called deposition. So that can affect the forest's health, and it can affect the forest's ability to provide the services that we depend on. For example, the forest provides us timber, it stabilizes our soil, it cleans our water. I have a picture of a stream here, and you can imagine that as the snow lands high in the mountains and melts, it comes down into our reservoirs, it's passing through these forest soils. So they are affecting our quality of life. This is a stand being marked for timber harvesting up in the Uintas. And it has been affected by bark beetles. Bark beetles are these bugs that drill into the bark of the trees and eat the cambium and make them die, which is a bummer. And it reduces the timber quality, and it also leaves all these dead trees that can catch on fire. And bark beetles and many other pathogens are exacerbated by poor air quality. So just as we've seen with people and the relationship between disease and air quality, forests have that same pattern. They are much more susceptible to other diseases when they are being stressed by poor air quality. Okay, so why lichens? You probably don't even really know what lichens are, but they're super cool and they live in forests. So if you go out and look at the trees in your front yard, you will see lichens. So here are some orange and brown lichens on a tree. This is a maple in Ogden. And here's a close-up of the orange lichens. Those are Xantho Mendoza. Here's a close-up of some of the brown lichens, Melanohalea. And some people think that these are harming the trees, but they're actually doing their own photosynthesis and living their own lives, and they just like happen to live on the trees. So they're not a tree disease or anything like that, but they have really interesting biology that relates to air pollution. If you look at a cross section of a lichen, so here's a cross section of this orange guy, you see this white layer on the bottom is fungus, and then this green layer is algae, and that's doing photosynthesis. And then this orange layer is a pigmented fungus. So these are two totally different species, fungus and an algae, living together, making this thing. And they don't have roots, and they don't have an ability to go into the tree. So what that means is they're getting all of their nutrients directly from the air. And if you take a cross section and throw it under a microscope, you'll see this layer of fungi, this layer of fungi up here, and then this little layer of algae. And the thing to notice is that unlike plants and people, lichens have no skin or cuticle to like protect them and make it so that pollutants can't come into their bodies. So they're basically sitting there like little sponges on the side of the tree, absorbing whatever is in the air, more or less in proportion to its abundance in the air. And that's why they're potentially interesting air pollution indicators. So they've been used in many different studies. This is an example from the Pacific Northwest where they're relating, this air score is calculated from lichens. and it's relating to the fine particulate nitrogen. And you can see the lichens are very strongly related to the fine particulate nitrogen. And then they can go out to bajillions of plots out on the land and look at the amounts of different lichens and interpolate how much nitrogen is landing in the forests. So that's what this map is here. And we can see places that are red might be having more nitrogen than is healthy for our forests landing in them. And here's another example from the literature. This is actually a meta-analysis that I did a few years ago. And it looks at how the amount of nitrogen in the lichens' bodies is related to the amount of nitrogen coming into the forest. And then all these different symbols are from different study areas. So overall, you see that there is a strong correlation that R squared is 0.58. But when you look at the different study areas, you can tell that they're not all the same. The relationship between what's in the lichens and what's coming into the forest is different depending on the study area. Okay, so what were my goals? I wanted to look at how this lichen nitrogen deposition were related in the forests in Utah. And then ultimately, I want to use that to make a map of nitrogen deposition in all the forests in Utah. So these are forest inventory plots that the Forest Service goes and measures regularly all across the state of Utah. They're on a 5-kilometer grid, and technicians go out and measure these every 10 years. So my thinking was, if they went and just like grabbed a few lichens when they were out there, and we knew how lichens were related to air quality, then we would have this beautiful map of how nitrogen was landing in our forests across the state. So that was the ultimate goal. All right, so let me tell you what we did to set this up. At different sites, I'll show you guys on a map in a minute, we set up these through-fall nitrogen deposition collectors. So let's look at these. These are under the trees, and nitrogen is coming into the forest through the rainfall and also through the dust and particulate matter landing on the tree's needles and being washed down into these collectors. And then at the same sites, we're collecting these lichens. This is one of my students, McKenna Hill, out collecting. She's carefully with gloves pulling the lichens off the trees and gathering them. And we're collecting a few different species, this orange one and also this brown one. And then the idea is once we correlate them, we could go anywhere and collect the lichens and be able to use this relationship to infer the through-fall nitrogen deposition. So we set up our sites in Utah up in the Uinta Mountains. You can see over here, kind of near Vernal, going in a north-south transect through the Uintas, sort of targeting the idea that there might be more nitrogen deposition associated with the development in the Uinta Basin. And we also set up sites in New Mexico near the San Juan Basin, which I'll focus on a little bit less, but they're part of the same study. And we went out and changed the through-fall collectors every fall and spring. And fortunately, we had 15 of them at each site because some of them had casualties. This one had a tree falling on it, which was a total bummer. And it was really, really hard to recollect the monitors. But anyway, we still had a bunch of samples out there, so it was okay. And then once we collected the through-fall deposition and the lichens, we sent them for ICP analysis, and we were able to get the nitrogen concentrations from both, as well as sulfur, carbon, a bunch of cations and heavy metals, which we're not going to talk about, but the same methods can be used for those. All right, so now we get to talk about what we found. All right, so since we collected the through-fall deposition every fall and spring, we were able to tell how much nitrogen landed in the forest in the winter versus the summer. So take a minute, all of you smart air quality people, and write down, do you think more nitrogen lands in the forests in the winter or in the summer? All right, are you sure? You're sure you know what you're talking about, okay? All right, I'm gonna show you the results. So I was super surprised by the results. I expected more nitrogen to land in the forests in the winter because that's when we have all these bad inversions and life is bad and there's all this pollution and that's not what happened. More nitrogen is landing in the summer in the forests. So over the time period, we're seeing this graph has the summer deposition, winter, summer, and winter. And this is in kilograms per hectare per month. And we're seeing quite a lot more nitrogen coming into the forest in the summer. And I think that that's because of the weather patterns carrying the air pollution and also the warmer temperatures making chemical reactions more likely to take place, which can bring the nitrogen out of the air and down into the forest. But I was pretty surprised by that. We can also look at the spatial pattern of through-fall deposition. So if you look at national models like CMAC and TDEP, these broad models predict that more nitrogen will land in forests at high elevation in this region, which is the opposite of what I actually measured. I measured more nitrogen deposition at the low elevation sites. So here's a map. You can see down here is the Uinta Basin, and all these little purple X's are oil and gas wells, and the green is agricultural lands. And then we have my plots, which are these pie charts. And so Little Mountain, which is near Vernal, and the first conifers on the Chipeta Lake Road, these two sites got the most nitrogen deposition. And then as you go up into the mountains, nitrogen deposition was lower. And then back on the north slope at the low elevation sites, it was higher again. And you can also see it was divided differently between reduced and oxidized nitrogen. Some of these sites, Little Mountain and First Conifers, and then the site over by Sheep Creek, and this site back here in the Deep Creek drainage, these were dominated by oxidized forms of nitrogen, whereas up in the mountains and by Elkhorn, these were more dominated by reduced forms. So that's what we found was coming into the forest. And now we want to relate that to what the lichens saw. So here we're looking at the lichen nitrogen content and the through-fall deposition coming into the forest. And now you might see why I'm not as interested in the New Mexico study area. That is symbolized by these circles. Basically, total failure there. The lichens were not related to the through-fall nitrogen deposition in my New Mexico study area. And I have some potential reasons why we'll get to in a minute. But in Utah, you can see that several of the species of lichens, so these are different kinds of lichens, did have positive relationships between the amount of nitrogen in the lichen and the deposition. And the R-squareds were between 0.31 and 0.58, which weren't fabulous. So I thought, okay, let's zero in on the season just before I sampled the lichens, and maybe that will give me a better estimate. So this graph here is for the average annual through-fall N. So that's over the 2 years of the study. And the next graph here is just for the winter preceding the lichen collection. And what we see is that the relationships are stronger. So here we have all the different species, and you can see this Usnea hirta. This is like a light green fluffy lichen. It's not a very good nitrogen bioindicator. But these other three are pretty good. And the best one was this Melanohalea exasperatula, which had an R-squared of 0.59, which is like, okay, but not great. So it's a reasonable bioindicator, but it could be potentially improved. All right, so what do we still want to know? Lots of things. One thing is we don't know where this deposition comes from. The lichens didn't tell us that. So that's something that other folks might need to help us with, or that we might need to look into in the future.
5 minutes.
Cool, thanks. Compared to other places where this technique has worked really well, our region has quite a lot more dry deposition. So this is nitrogen coming into the forest in the form of dust. And in other places where this method has worked, most of the nitrogen coming into the forest has been dissolved in rainfall. And we might expect that lichens would absorb nitrogen differently. If it's dissolved in rainfall, it might be easier for the lichens to take into their bodies than if it's in fine versus coarse dust particles. And some folks in Italy have been studying this. This is some of their SEM work looking at the particle size. So we still need to understand how lichens are absorbing nitrogen from different kinds of particles. We had lots of different forests. A lot of places where this has been a really successful method are basically like all big Douglas fir forests or similar forests. Whereas in Utah, when you go up and down the mountains, you're dealing with like pinyon pines and junipers down at the low elevations, and then these big spruces and firs at the high elevations. And that's going to change how the nitrogen comes into the forest through the canopy. So that's adding variability. So we didn't have success in New Mexico, and I have some ideas why. When sampling happened there, it was in between all these crazy flashy thunderstorms with lots of rain coming all at once. And I think that that could affect how the lichens are absorbing the nitrogen in terms of leaching out the nitrogens from their bodies. The technicians also didn't collect the throughfall deposition and the lichens at the same time. So we can't zero in on just the season right before the lichens were collected like we did in Utah. And then within the plots, there was quite a lot of variability in lichen collections. So I sampled multiple lichens in the plots to make sure that they were consistent. And in Utah, they were super consistent. And in New Mexico, they were not so much. All right, so lichens can tell us about spatial patterns of deposition. In Utah, we found a reasonable correlation. We can use this to map how nitrogen is coming into the forests, at least in some areas. But there are limitations. This method was not successful in New Mexico. It integrates over time, so this isn't going to tell us about specific inversion events. The lichens are taking these nutrients into their bodies over multiple months. We might be able to improve this method by focusing on the species that worked best, like the Melanohalia. Making sure that we time sample collection when there's not like major thunderstorms that might affect our results. And another approach might be after we sample the lichens to rinse them off in water and separate the amount of nitrogen that's just settled on dust on top of them versus actually absorbed into their tissues. And I think that that's an important place for this to go next. So I want to acknowledge the Forest Service. They provided all of the funding for this through their inventory and analysis program. And I had tons of great help from Weber State students in particular. And I'm thankful to you guys for listening, and I'm happy to take questions.
Great. Thanks so much, Heather. We have time for a question.
So, you mentioned or showed that there is a seasonal variability there that implies a biological half-life. How long do you think this will hold a sample?
What do you mean?
Sorry about that. So in other words, when this absorbs some nitrogen and then you measure it, are you measuring nitrogen from the last month or two or from the last several years slowly decaying? So how long does the nitrogen that you're measuring stay in the lichen before you get a fresh reading, so to speak?
That's a good question. And transplant studies have looked at that. So if you like take a lichen from a clean place and put it in a more polluted place, it usually takes a couple months to reflect the pollution in the place that it's been. So this is on it, and it depends on how fast they're growing, what their lives are like. So it's a few months. It's not, you couldn't pin it down to like a number of days, but it's on the order of a few months.
Great, thanks. There's kind of a similar question and some side questions in the chat. So if you have time to look at that, Heather, that'd be great. And we better move on to the next talk. Thanks again.